My name is Rita Pinho and I'm a third-year PhD student here at the UCL Institute of Ophthalmology. I usually define myself as an engineer by training and biologist by heart.
My name's Louise Morgan and I'm a research associate in Julie Daniels' group at the Institute of Ophthalmology at University College London. My job is to develop a product that has been invented by Julie Daniels and other collaborators that is a compressed membrane of collagen that can be used to deliver stem cells to the front of the eye. Using natural products, people have been able to deliver stem cells and replace them and then get a fantastic improvement in the front of the eye. The clarity comes back and the pain goes. But they use a natural product that's very hard to get and very unreproducible. So the point of the project is to develop this tissue equivalent to such an extent that you can have an easily available and very reproducible product that can be available to anyone.
So my project stands between the basic biology and the translational clinical side of it. So the first part is to find the best localization to isolate corneal stromal stem cells that are the progenitor cells of keratocytes, that are the cells that live in the stroma, that is the thickest layer of the cornea. And the second part is a way of delivering these cells to patients, and for that we use our RAF tissue equivalents, that is a collagen compressed hydrogel that has been developed in this lab for the past 10 years. UCL is a world-leading university and London is an amazing city, but what really persuaded me to come to this lab was the translational side of it. Since we are close to Moorfields Eye Hospital and we have the stem cell therapy unit, we develop real therapies for real patients, so that seemed quite exciting. So in my opinion, there are three key points about being here. That is the close proximity with the hospital and the eye bank that gives us access to human tissue that is key for our research. The fact that we have the opportunity to learn about regulations and how to develop a product under GMP conditions, and the freedom that Julie gives us to explore new avenues and get involved in different projects, even if it is public engagement or teaching.
Well, most of my career I've worked within a drug company and I was looking at new targets for drugs, and it was great fun and very nice, but you spend a lot of time looking at things that you know are never going to go anywhere. So when I came to Julie Daniels' lab, I'd looked at for a place where I could do something that was actually nearer the end point. And Julie Daniels has a good reputation as a scientist looking at the stem cells in this particular area, and I thought there was a good chance of being able to work at something that could actually end up being a product that could be used. And when I came and looked at the place here and looked at for interview, I saw that actually not only was that the case, she has a fantastic group, different people doing from all sorts of different, people from engineering, people from regulatory affairs, very good scientists. I thought it would be exciting, and a lot of clinicians that they were collaborating with. I thought it brought together all the things that I was slightly a few steps away from in the earlier parts of my career. Within Julie Daniels' lab, we have people who work at the early stage of the science, but also we have a group of people who are ready to develop it in the clean room, who are discussing with clinicians and with regulatory affairs. So they will come, we'll have group meetings, and when we say we want to do this, they'll say, well, do it this way, so that we don't have to repeat things when we come to produce things ready for the clinic. So very early we'll be considering what we need at the end.
So this project is of extreme importance because we have proved that corneal stromal stem cells support the growth of limbal epithelial stem cells, that are the cells of the outer surface of the cornea.
So RAFT, if it does everything we want it to do, the cells will grow on it just as well as on amnion, and they will be delivered to the eye and cause no problem in the eye when they're there. And the surgeons will have a product almost on the shelf ready to use. They won't have to go looking for a donor and characterizing. The challenges in developing RAFT have been we know we can get the RAFT itself developed in a very nice reproducible manner. We've worked with the company that has given us we call it a plunger that will compress our RAFT, and it's very nice reproducible system. When we add it to the cells, we have to use human donor tissue to get what we're going to be using. and most of the problems has been in getting a good standard to ensure that we have a standard procedure, given every donor is different and every prep of cells that we use is different. The other thing we've been doing is to check that it's safe. So we have done a lot of safety studies as well, which have actually panned out in the end very well, and we think we have a product that is safe.
So we hope that in a couple of years we'll have the first in man and raft being successfully implanted into humans, hoping to alleviate blindness worldwide.
Well, it would be very exciting to see the product go into the clinic. We would love to be around and still in the lab when the first person actually receives raft, and we know that we have actually delivered a new product, a new choice for surgeons to use when they're treating these very painful and blinding but debilitating diseases.